engineering Saccharomyces cerevisiae to deliver targeted dsRNA against Varroa might via honeybee hosts. Hi, I'm Parisa. I'll begin by introducing the problem we're addressing. Varroa mites is one of the biggest global threat to honeybee health. It spreads deadly viruses and weakens bee immunity and often leading to colony collapse. Traditionally, beekeepers have used chemical acaricides, but over time these have caused major issues. Mites developed resistance, chemical residue, contaminated honey in bags, and the treatment themselves harm the bees. To offer a more targeted and sustainable approach, Bertolin and colleagues in 2025 trialed an RNAi-based strategy. They fed bees dsRNA targeting key mite genes, which successfully led to gene silencing in the mites. Compared to chemical pesticides, RNAi offers greater precision, fewer toxic residue, and less risk of resistance. Our aim is in this project is to engineer Saccharomyces cerevisiae that produce dsRNA inside the bee's gut, providing a more sustainable and hands-free delivery method. We selected three essential mitogens based on the Bertolin study and designed plasmid carrying key genetic components. We used bioinformatic informatic tools to optimize the sequences and ensure expression compatibility with yeast. We also selected a yeast strain updated to high temperature in gut condition, aiming to a stable colonization or effective lysis inside the bee. The approach brings key advantages continues in vivo dsRNA production, eliminating the need for the repeated feeding and enhancing stability. And however, challenges remain, such as ensuring efficient yeast lysis dsRNA uptake by mice and minimizing off-target effects. Overall, this strategy lays a strong foundation for sustainable mite control. Finally, our designs support multiple SDGs, SDG2 by protecting pollen, pollinator and securing food system, 3 by avoiding chemical residue, 12 by enabling localized in hive production, 13 by enhancing ecological resilience, and 15 by promoting biodiversity with targeted pest control.
So in terms of our mechanism and molecular pathway of our design, we will be inserting a strain of Saccharomyces cerevisiae yeast that is capable of surviving in the midgut of a honeybee into the bee's microbiome. And this yeast cell will be engineered, has been engineered and designed to produce single-stranded RNA, which, when converted to small interference RNA, is capable of silencing the protein expression of proteins that are critical for Varroa mite survival. Um, so how does this single-stranded RNA become small interference RNA? Well, to do that, we've also inserted a gene for RNA-dependent RNA polymerase into the Saccharomyces cerevisiae. Um, and this is sourced from another fungal species called Neurospora crassa, and this is able to produce double-stranded RNA within the cell. So when the Varroa mites parasitize the honeybee, the double-stranded RNA is uptaken into the Varroa mite, where it binds to a dicer protein. This dicer protein converts it into small RNA, which produces an RNA-induced silencing complex, which is capable of binding and cleaving mRNA, which encode proteins critical to Varroa mite survival. These targets include endochitinase, which is important for uh, exoskeleton growth and molting, sodium potassium ATPase pumps, which are important for maintaining ion gradients, and acetyl-CoA carboxylase, which is important for synthesis of fatty acids. All of these are important for the survival of the Varroa mites, and by cleaving them, we can essentially kill these mites. and protect the bee.
So as mentioned, the organism selected for engineering is Saccharomyces cerevisiae, specifically the ATCC 9080 strand. This strand has been identified in the honeybee gut microbiome as demonstrated by Argabetti et al., indicating its potential for survival and functionality within the hive environment. For gene delivery, the plasmid PRS414 will be used as it is a low-copy yeast-compatible vector. The synthetic construct will include the QDE1 gene, which encodes an RNA-dependent polymerase. This enzyme will catalyze the formation of double-stranded RNA from transcript targeting the essential Varroa mite genes. These genes include the endochitinase, sodium-potassium ion ATPase, and acetyl-CoA carboxylase. The T7 promoter will initiate transcription with an RNA-dependent polymerase, which will use the mRNA of those targeted genes to synthesize double-stranded RNA. The CPS1 terminator will signal for the end of transcription. PSP-OMI and NOTI will act as restriction sites. Finally, the AMPR genes is used to ensure the yeast has taken out the plasmid. We are targeting the genes of three enzymes mentioned above: endochitinase, sodium potassium ion ATPase, and acetyl-CoA carboxylase. By silencing the endochitinase gene, the Varroa mite's cuticle integrity and digestive system will be disrupted, leading to interference with development and survival. The the sodium potassium ATPase is essential for the mice nervous system and cellular homeostasis. Targeting it will cause cellular dysfunction and ultimately death to the mice. And lastly, the acetyl-CoA carboxylase is involved in fatty acid biosynthesis, energy storage, and membrane formation. By targeting this enzyme, the mice reproduction will be disrupted. Yeah.
So this is a result of like plasmic and then the cut block of diagram of genetic parts. So we use like Gibson assembly, so which will effectively join multiple DNA fragments together via PCR with Brammer. So here you can see the cutting block. We have like the QDE1 gene with three target might gene with like the T7 promoter and S1 terminator. And this is a demonstration plasmid diagram. And because like it will need to use like Gibson assembly, so this is our assembly primer. We construct like BRS414 forward and reverse primer and board primer for like three gene terminator as well as like QDE1. And here's the parameter if we need to use in like the lab control.
We are talking about the testing after successfully engineering our Saccharomyces cerevisiae strain with the plasmid, carrying our dsRNA constructs. We conducted a series of validation experiments under a control condition before moving into the field testing. So we began with the restriction digest analysis using the PSPOMI and NOTI enzymes. These enzymes got the plasmid at the known locations, allowing us to verify the presence and correct insertion of our target gene sequence. The results were visualized through the gel electrophoresis. So if you look at this gel image, lane 1 shows two clear DNA bands. The upper band at around 5,500 base pairs correspond to the three targeted Varroa uh, genes plus the QDE1 gene. The lower band at about 4,700 base pair represent the plasmid backbone. These band sizes match our expectation and confirm that the plasmid was successfully constructed. To further validate the construct, we performed the PCR amplification using the six sets of specific primers targeting VDACC, VDCSIT, BDATPs, and QDE1. These ensure that all the necessary sequences were present in the transformed yeast. We then carried out RT-PCR to confirm the actual expression, checking at the engineered yeast is actively producing dsRNA from our inserted gene. So finally, we conducted the stability and efficiency testing by stimulating honeybee gut condition using the qPCR and gel-based assay. We measured how stable the dsRNA remains over time and whether it stays intact long enough to positively interfere with the Varroa gene. So, yeah, together these steps confirm that our engineer yeast is not only genetically accurate but also functionally viable. Uh, yeah.
As we look towards the future of our project, the next critical step is testing our engineered genes in real honey colonies. Apart from the laboratory showing promise, field testing is essential and how the yeast behave with the complex environment of a living hive in the beehive. We'll begin by examining the stability of the yeast in bee gut, ensuring it's survived long enough to produce and release the double-stranded RNA. Using qPCR, will quantify yeast level over time and verify whether successful dRNA is produced and absorbed in the bee system or not. Most importantly, we will monitor whether the RNAi interference effectively reduces varroites population, specifically by targeting critical genes such as ACC, chitokinase, and ATPase. Beyond the mites, we're equally concerned with the bee themselves. We'll assemble the bee mortality, colony strength, foraging behavior to make sure our solution is safe and doesn't harm the host organism as well. These steps are crucial for translating lab success in the real world impact. In conclusion, this project represents a sustainable breakthrough in pest management through synthetic biology by engineering yeast to produce dRNAs inside the bee, and we aim to overcome the deliberate challenge that held DNA technology back. Our approach offers a high targeted, bee friendly, and ecologically responsible alternative to the chemical pest present in the modern world. If successful, this strategy could reshape how we protect pollinators not only in Australia but all over the global advancement level, while advancing a new era of symbiotic technological agricultural impact can be shown from this project. Thank you so much. And these are currently the reference that we use from some paper to present our paper and project. Thank you.